What if I told you we could reach into a brain, find the exact cells that hold a single memory, and then, with just a flash of light, change it completely? Sounds like pure science fiction, right? Well, today we're going to break down the incredible experiment that actually made it happen. You know, we all like to think our memory is this perfect, high-definition recording of our past. But we know that's not really true, don't we? We forget names, we get details mixed up. We've all been in that argument with someone over how an event really went down. Our memories, well, they have glitches. Just think about that for a second. That feeling of being totally 100% sure about something, only to have the rug pulled out from under you when someone shows you it didn't happen that way at all. It's a weird, disorienting feeling, and it really shows you just how flexible our memories can be. Okay, so we know memories can be flawed, they can be unreliable, but that leads to a much bigger, much wilder question. Is it possible to have a vivid emotional memory of something that just never happened? That's the exact rabbit hole a team of neuroscientists decided to go down. Now, to even think about trying an experiment like this, you need some seriously powerful tools. And the key to this whole thing is a mind-blowing technology that lets scientists target one specific memory with, like, surgical precision. So first, we gotta talk about something called a memory engram. Basically, when you form a memory, let's say of this room you're in right now, a very specific little group of neurons in your brain fires up. That unique pattern of cells is the engram. It's the physical trace of the memory. The best way to think of it is like the brain's dedicated save file for that one experience. And this is where things get really wild. There's a technique called optogenetics. It lets scientists genetically tweak neurons so that they respond to light. They're basically installing a tiny light-activated on-off switch directly into the exact brain cells they want to control. It's incredible. And this image here shows you how they actually do it. They use a harmless virus to carry that genetic light switch into a mouse's hippocampus. That's a super important area for memory. Then they implant this tiny little optical fiber so they can beam pulses of blue light right onto those cells whenever they want. So you might be wondering, how do they get that light switch into only the right cells, into the engram for just one specific memory? Well, the system is just brilliant. They let a mouse explore a new room. As the mouse is building the memory of that place, only the active neurons, the ones forming the engram, get tagged with that light switch. And just like that, the scientist can now turn that one specific memory on and off just by flicking a light switch. So with all this incredible tech ready to go, the researchers came up with this really elegant 3-day experiment. You can almost think of it as a kind of memory heist. But the goal here isn't to steal a memory, it's to plant one. This right here, this is the blueprint for the whole operation. I know it looks a little complicated, but it really just breaks down into 3 simple days: label, condition, and test. All right, day 1. We'll call this the label. A mouse just explores a totally normal, safe room. Let's call it context A. While it's sniffing around, the neurons that are storing the memory of context A get tagged with that light switch. That's it. The save file for the safe room is now labeled and ready to go. Day 2 is the big one. This is the crucial move. The mouse is put into a completely different room, context B. And in this room, it gets a mild, unpleasant little foot shock. But here's the crazy twist. At the very same moment it gets the shock, the scientists shine that blue light into its brain, artificially turning on the memory of the safe room, context A. Think about that. The mouse is feeling fear, but it's being forced to recall a memory of a place where absolutely nothing bad ever happened. And all of that leads us to the final make or break test on day 3. The mouse is put back into context A, the original safe room. And remember, it was never shocked in this room. Not once. The only link between this room and a feeling of fear is the artificial one the scientists created. So what do you think happened? Did it work? Did they actually succeed in planting a false memory? Was this mouse now afraid of a place that should have felt completely safe? Well, the results were absolutely stunning. They gave us the first real, direct proof that you could implant a complex false memory and have it show up as actual, real behavior. Okay. Just look at the data here. In mice, a classic sign of fear is that they freeze. They just stop moving completely. This chart shows how much time the mice spent freezing when they were put back in that safe room. The control mice, they barely froze at all, less than 5% of the time. But the mice with the fake memory, They were frozen solid for about 30% of the time. They were genuinely terrified of a place where, I'll say it again, nothing bad had ever happened to them. You don't have to take my word for it. The scientists themselves said it best in their paper. They proved it was possible to artificially create a fear memory that wasn't just some blip in the brain. It was a real behavioral thing. To that mouse, the false memory was completely real. So why did this work so incredibly well? Well, turns out, in the brain, location is everything. The experiment was a success in a part of the hippocampus called the dentate gyrus, or DG. In the DG, the save files for different places are super distinct, with almost no overlap. When they tried the exact same experiment in another nearby area, the CA1, it failed. Why? Because in CA1, the memory codes are all jumbled together, much more overlapped. The signal was just too noisy to cleanly implant the false link. It just goes to show you how ridiculously precise our memory system can be. So, okay, scientists can plant a false memory in a mouse. It's a really cool scientific achievement, for sure. But what does it actually mean for us, outside of a lab? Well, the implications are absolutely huge. First, this pretty much confirms that your memory isn't a video camera. It's a creative process, a reconstruction that can be edited. Now, on the one hand, this could open the door to amazing new therapies. I mean, imagine being able to help someone with PTSD by turning down the volume on a traumatic memory. But on the other hand, this raises some really serious ethical red flags. Our entire legal system, for instance, kind of depends on the idea that an eyewitness testimony is a reliable record of what happened. This research basically throws a giant wrench into that idea. And that really leaves us with one final, pretty unsettling question to chew on. Our memories, they define who we are. They are literally the story of our lives. But if that story can be edited, if our memories can be written and rewritten, who should get to hold the pen?